to provide a platform to the scholar, teachers, and experts from various fields of science and technology for academic interaction, elevation, and enrichment. It has also taken initiative in helping students in extending their mind to diverse fields available in biological science and bioinformatics. This year, we have organized a national webinar on the topic of fungal genomics and its implications. Now, I would request Principal Madam Dr. Pratibha Gaikwad to address the participant.
Thank you, madam. A very good morning to one and all. On behalf of Chikitsak Samuha's Patkar-Varde College, it gives me immense pleasure to place on record my high level of appreciation towards today's two resource person, Honorable Dr. Shenoy sir and Honorable Dr. Vijaya madam. I also congratulate the Bioinformatics department of Patkar-Varde College, who is run under the efficient administration of the CEO, Honorable Dr. Mala Kharkar Madam, who regularly conducts this best practice of organizing national seminars, SCIMATIX. Today's seminar is on fungal genomics, and this is a very important topic, especially for the students. Applicability of science plays an important role in bettering the mankind, and secondly, becoming job employable or having the capacity to earn a living. So this particular seminar would be of great importance for all the students, because this area is of great importance for the betterment of the life of the students. So with the advent of National Education Policy 2020, right from the beginning, Patkar-Varde College's Bioinformatics department has always been instrumental in providing applicable knowledge to the students under the eminent administration and leadership of Dr. Ruchita Dalvi madam. I am very happy to appreciate her efforts in organizing this national seminar, and certainly this seminar will add to the existing branch of knowledge and will enlighten the students in the right direction, right path, which they have to adopt after obtaining their graduation degree. So with these few words, on behalf of the management of Patkar-Varde College, to name a few, our mentor and guide, Honorable Mr. Rangnekar sir, the President of Chikitsak Samuha, the very dynamic Joint Secretary of Chikitsak Samuha, Honorable Dr. Gurunath Pandit sir, and our own very much with us all the time, day in and day out, the CEO of Patkar-Varde College, Dr. Mala Kharkar madam, and myself, the principal, takes great pride and privilege to welcome all, especially the two resource persons, and I wish you all a very fruitful and meaningful activity throughout the day through this national webinar. Once again, I appreciate the efforts of the department, the team, and I thank the resource person for sparing their valuable time and making it possible to address this very enthusiastic crowd who will be the future leaders of India. With these few words, I declare this webinar to be open, and I wish you all the best. Thank you. Jai Hind, Jai Maharashtra.
Thank you so much, ma'am. Today, we also have among us the co-founder of MicroAsia, that is Dr. Shenoy. Sir is a fungal taxonomist by training. After completing his bachelor and master degree from India, Sir has pursued his PhD in fungal taxonomy at University of Hong Kong, China. Thesis title, Multigene Phylogeny of Selected Anamorphic Ascomites, dealt with evolutionary relationship of asexual genera and provide insight into implication of the polyphyly in the fungal taxonomy. Sir has worked as a postdoctorate fellow at the Agriculture and Agri-Food Canada, Ottawa, and in DNA barcoding of Fuserum. In 2008, Dr. Shenoy returned to India and joined as a fungal taxonomy at microbial type culture collection at CSR Institute of Microbial Technology, Chandigarh. He has supervised two PhD students there and published several high impact papers on fungal taxonomy, phylogeny, DNA barcoding, and comparative genomes. In 2013, Dr. Shenoy moved to CSR, National Institute of Oceanography, Goa, and initiated research on diversity and taxonomy of microbes associated with star balls. Dr. Shenoy is presently working at CSR, National Institute of Oceanography Regional Center, Vishakhapatnam, Andhra Pradesh, and has supervised two PhD students on bacterial diversity on select marine natures. Dr. Shenoy has so far published 45 papers in reputed international journals. He is co-founder and presently the managing editor of a new mycology journal, MycoAsia Journal of Modern Mycology. Recently, he has founded a new mycology journal for Indian fungi, MycoIndia, MycoIndia Journal of Indian Fungi. Dr. Shenoy has been a member of International Subcommission on Collectotrichum Taxonomy. Dr. Shenoy is on the advisory board of MIT Institute of Oceanography and Atmospheric Science, India. It is an honor to be with you, sir. We welcome you, sir, for this webinar. Now I request sir to address the participant.
Uh, Good morning, ladies and gentlemen. I hope I am audible.
Yes, sir.
Thank you. First of all, thank you very much, madam, for introducing me to the today's audience. Thank you very much. I'd like to thank Dr. Ruchika Dalvi and team at the Patkar-Varde College, Mumbai, for collaborating with MycoAsia for today's event. Um, Talking about MycoAsia, we started this journal in 2019. There are 5 co-founders and 1 advisor. 3 co-founders are from Sri Lanka. 2 co-founders are from India, including me. We started as a journal. Along the way, we have learned 2 lessons. Number 1, we should focus on quality over quantity, actually. Number 2, we should not be just a journal. We should be helping people learn new things, latest information. So in that pursuit, we have been conducting online training programs, sometimes offline interactions. So during this kind of interaction, I believe Dr. Ruchita Dalve came in contact with us, and she kind of discussed with us the possibility of arranging this kind of webinar. So during the course of discussion, we requested Dr. Vijaya Brahma, who is today's speaker, to kindly, consider being the speaker for today's event. Actually, she has been very kind and she has agreed to be the today's, expert. And I'm happy that MycoAsia and Patkar-Varde College is collaborating, and hopefully we are going to have a wonderful session on fungal genomics and its implications. And I see many people in the audience who are the friends of MycoAsia. I thank all of them for joining today. I thank Vijaya Brahma for accepting the role as the expert today. Thank you, Vijaya. Thank you very much. And Dr. Ruchita Dalvi, thank you very much again. With this, I hand over the platform to the madam who has introduced me to today's audience.
Thank you, sir. Thank you so much.
Thank you.
Moving ahead with the session, in today's webinar, we will highlight on the topics on fungal genome and its implication. So let me introduce something regarding to that particular topic. Fungal genome is a scientific discipline that concerns the genomes encircling the entire hereditary information on fungi. The genomes of fungi encode genes that enables them to thrive in diverse environment in which plant and animal cell and participate in nutrient cycling in the terrestrial and aquatic ecosystem. The continuously expanding database of fungal genome sequence have been generated by individual. This genomic database can be utilized to better understand how fungi have adapted to their lifestyle and ecologic niches. Large data set of fungal genomic and transcriptomic data has enabled the use of novel methodology and improved the study of fungal evolution from a molecular sequence perspective. I extend a warm welcome to our speaker for today's webinar, Freelance Project Manager, Dr. Vijaya Brahma, who will enlighten us with this topic. Before that, I would like to introduce you all to our speaker over here. Dr. Vijaya Brahma holds a doctorate in microbiome research with a focus on soil metagenomes. She completed her PhD from CSR MTech in Chandigarh. Following her doctorate study, Dr. Brahma pursued a postdoctorate fellowship at the University of Toronto, where she contributed in the hospital research project on lung microbiome. Her research interest led her to join SCIS JNU as a UG postdoctorate fellow specialized in plant transcriptomics. Throughout her career, Dr. Brahma had gained diverse research experience. She served as a research associate at GATI, a DST project under Kiran Division, where she contributed to significant scientific advancement. Additionally, she worked as a mentor with NGOs Vigyanshala International, sharing her knowledge and passion for science. Dr. Brahma successfully transitioned into a project management role, contributing her expertise to organizations such as Atolo Social Enterprise in Canada and the Freedom Employability Academy. Most recently, she has been associated with Ubuntu, where she continued to make a positive impact as a member of esteemed scientific society like the American and Canada Society of Microbiology. Dr. Brahma brings a wealth of knowledge and expertise to her field. Her areas of specialization include bioinformatics, data analysis, scientific communication, statistics, project management, and programming skills. Dr. Brahma contribution to the society and scientific community are evident in her impressive publication record with 15 international peer-reviewed publications to her name. Her dedication and innovation have also been recognized with USA patent in her field. With her strong academic background, research experience, and expertise in various domains, Dr. Vijaya Brahma continued to excel in the field of microbiome research, making valuable contribution to scientific advancement and interdisciplinary collaboration. It is an honor to have you, ma'am, with us. Welcome, ma'am, for the seminar over here. Before we begin the webinar, a few instructions to the participant. I request all the participants to kindly be patient if any technical problem arises during the session, which we'll try to solve all the technical glitches. Kindly mute the audio and video for the smooth conductance of the session. Kindly post your question and a doubt in the chat box. It will be answered at the end of the session. Ma'am, I request you to begin the session. Over to you, ma'am. Thank you so much.
Thank you for this lovely introduction. And I have heartfelt gratitude to Patkar-Varde College for giving me this opportunity. I would say that you define the fungal genomics far better than the way I was trying to define, because I thought that I'll make it very simple and very preliminary for everyone to understand. And special thanks to the Honorable Principal Ma'am, the Bioinformatics Department, Ruchika Dalvi, and the Microasia, of course, for giving this wonderful opportunity. It's always a pleasure to present the learnings and then share the knowledge. We are constantly in a learning process, and we would definitely learn together. So without further ado, I'm going to share my screen now. Do confirm that the screen is visible now.
Yes.
Thank you very much. So the topic is fungal genomics and its implication. This is a lecture by me. My name is Vijaya Brahma. I was fortunate enough to get my hands dirty with the microbiome and metagenome, and it was really, really the hot topic in the early 90s and 2000s when the technology was just starting, and I was fortunate to be in a lab where we could afford these kind of sequencing work and present my work nationally and internationally. So with whatever learning I have, I'm going to present to you. Just heads up is that I am not a fungal genomics expert per se, but I have done a lot of genomics and transcriptomics related work. So it applies to all biological systems. Special thanks to the Department of Bioinformatics, Microasia, and the RUSA sponsored national webinar. So the fungal genomics is a study of the entire genome of an organism. In this case, it's a fungi. Fungi are the well-established model organisms for research. They have several advantages over other model organisms because they are relatively simple, easy to grow, manipulate in the laboratory. They have well-established genetic toolkit like gene knockout system, gene tagging, whole genome sequencing for that matter. And they're closely related to humans, sharing many of the same genes and the biological processes. So before we move ahead, we should have this thing established that how genomics is different from genetics. So genetics is the traditional method of studying the hereditary traits of an organism and their variations within the population. On contrary, genomics is a study of genomes, a complete set of genetic material of the organism. The genetics was introduced by Groger Mendel in 1865, which focuses on the behavior of the genes, and we use employ different techniques like biochemistry and biology to study the single gene. Genomics, on the other hand, took a wave in the late 80s. The term was coined by Tom Roderick in 1986. It focuses on the entire genome of the organism. It can be studied with the use of bioinformatics, molecular biology, and it also involves a study of the interaction of the genes. I just put this other slide, which is a very small preliminary introductory slide for the people who are at the very basic level of these studies, and giving the definition of what is a gene, genome, chromosomes, and DNA. Let's move ahead. So this is an entire roadmap of the milestones which have been done so far in genome assembly. So the timeline illustrating many of the major genome assembly achievements ranging from beginning of the sequencing era, which is 1965, which was like the small shotgun sequencing, the traditional sequencing methods, to large scale genome projects, which are currently ongoing. So this is still till 2020, but we know that still major big research projects are going on. Big consortiums are doing sequencing projects, and there is no end to it. It's still a booming subject. Each genome or each genome project is placed under a color code in the background. If you see, the light red are the early sequencing methods, yellow being the Sanger shotgun sequencing, and green is the next generation sequencing. But light green are the modern, next to the next generation sequencing is the third generation sequencing techniques, which are much more longer reads, much more error proof, and much more like more high technologically advanced sequencing reads. So there are different areas of genomics, and we will touch each area one by one. Before that, I also wanted to share this a very interesting story of, I'll do a little bit of storytelling, please bear with me, because it builds the whole excitement about the sequencing, that how early in 80s, when nobody could even think of like sequencing something, come up with whole sequencing an entire human genome per se. So this is a story about Thomas Roderick, the person who coined the term genomics. He was born in 1930s, I guess, and he was a geneticist. He was in University of Michigan, Jackson Laboratory, and worked in behavioral genetics. So what he was studying was the radiation in the genetic material and also impact of health on the nuclear radiation. While discussing the feasibility of sequencing the genome, that time they were thinking of sequencing the human genome. It was like sitting in the bar around 1986. Around the sequencing was pretty much established with the smaller sequences like phage sequencing and bacterial sequencings had been established. For that matter, Saccharomyces cerevisiae was also around that time booming and had been sequenced. So they were thinking of mapping the entire genome, the human genome. And while they were thinking about mapping the entire human genome, the word genomics came into their mind. And that was the day when the genomics terms was coined. And finally, the project was launched. And by 2003, the first draft of human genome was published. It was a big thing. It was a really big thing, sequencing the entire human genome. And eventually, bigger and bigger and better advances have been taking place. But we cannot undermine the sequencing of the model organisms, which give a lot of depth of information to us about the human genome, because there are a lot of sequence similarities in them. For that matter, the fungal genomes like Saccharomyces cerevisiae and other genomes. So we will be touching each area. I have pointed out 10 areas mainly, but genomics per se is only comparative, functional, and structural genomics. But there are many super specializations in this area where you can explore your genomic data once you have it into different areas. And it's all the matter of what kind of questions you're asking and what kind of problem you want to resolve. And based on that, you can maneuver your genomic sequencing data into different areas of specialization and ask a right question about it. So starting with the genome sequence. While I start, I just want to give you a heads up that this will be only a cursory high-level overview of the topic, because each topic here is an independent seminar on its own. There is a lot of depth, a lot of research happened over the years. So I will be doing justice for the sake of respecting the time as well. And it will be very cursory level of giving you a tip of the iceberg feel to the different areas of research, and we can always delve deeper. I can come back again, or you can also have extensive research articles and read deeper into it, and feel free to email me if you want some resources as well in the future. So the whole genome sequencing was the most powerful and empowering thing that happened in the area of genomics, because one could precisely determine the genome sequence through more efficient technologies characterized by increased accuracy, throughput, and sequencing speed. And the cost was also dropped drastically. It was affordable to do whole genome sequencing. So the typical sequencing flow of next generation sequencing usually is that you extract the genomic DNA. It could be any source. Once the sequencing is extracted, there are established protocols of having these protocols to extract the DNA. Once it is done, you do the shearing. Shearing is like breaking down into smaller pieces. There are different methods for this: nebulization, sonication, and the end fragments are repaired. Once the end fragments are repaired, then ligation is done with the adapters. When you are doing next generation sequencing, there are special barcodes which are attached to it, because you have to do it in a high throughput parallel sequencing, multiplex sequencing. Then these are created into libraries where purification is done through streptavidin or magnetic bead method, and the library is amplified. Now, this is a different companies have a different chemistry going on in amplification, like emulsion PCR in the Roche Torrent, or bridge amplification happening in the Illumina generation. Once you have the sequence, it's just the first part. You just have the sequence, and the assembly begins. The assembly of the sequence, the gene annotation part of it, and then you can, as I said, that what kind of question you're asking. Based on that, you do the further analyses based on the SNP calling, variant analysis, or whatever is the requirement of the purpose of the sequencing sample. So next generation genomic technology collect increased amount of genomic data. we know that there's enormous data lying there, and it's been, for that matter, NCBI, the data repository, has already crossed more than 50 years. So it has a millions and millions base pairs of data. So that's another problem on its own, which is like a big data problem, where how much data you have and what can you do with that data, and how to utilize that data. So even if a small laboratory who does not have fund enough to do a sequencing per se, they can always have those repositories and ask right kind of questions and still have churn out a lot of information from the submitted repository of data. So I tried to put 10 of the most prominent fungal genomes which has been sequenced so far. If bear with me, if the screen is too small for you, probably you can, if you're on a phone, you can like zoom in and see. So there's Saccharomyces cerevisiae, which is the baker yeast, Candida albicans, Aspergillus fumigatus, Fusarium chrysosporium, Magniportha oryzae, Cryptococcus neoformans, Neurospora crassa. Then there's Coccydus immitis and Penicillium chrysogenium and Rhizopus stolonifer. So these are just to name few, but these all have an economic importance. Some are important in disease, some are important in plant and crop pathogenesis, and a lot of information has been shed light upon by the genomic data. So it has significantly accelerated, making it possible to sequence the entire genome at a lower cost, as I said. But I want to point your notice into two organizations which are extensively working in genomics, which is one is the Broad Institute, which is the Fungal Genomics Institute, which is dedicating entire fungal genomes, and there's a repository dedicated to the fungal genomes. They aim to sequence like many, like more than 1,000 genomes. Another one is the JGI, which is a Joint Genome Institute. They have this microcosm, which is a dedicated repository where you can do a lot of annotations, and a lot of genomes are there to download, and a lot of downstream pipeline sequencing analysis can be done in these repositories. So the Fungal Genomics Institute has committed to sequence like 10,000 fungal species by 2030, to annotate them to high level of detail. When I say annotation, it's not only computational annotation. The annotation has to be cross-validated through wet lab as well. So these genomes are annotated, and these genomes are species are available to public through centralized databases. So the kind of thing which even great causes like Mycoacia are doing, they're making information accessible to all. So the accessibility is very, very crucial in increasing the reachability of information, and to promote the use of fungal genomes in research and discovery. So Broad Institute is highly working on it. If somebody wants to pursue further research, they can always write to the scientists, and they are very, very cooperative in exchange of information regarding the genome sequences related to fungi, any troubleshootings. They are very, very kind to respond. The next is the JGI, which is 1,000 fungal genomes. And let me zoom if I can. Yes. So the Microcosm web portal provides the data access. There are visualization tools to see the data, and analysis tools for comparative genomics of fungi. I will explain more about comparative genomics in the further slides. So it enables the user to navigate through the sequencing fungal genomes and to conduct comparative genome-centric analysis of fungi, and community annotation. So as I said, that a power of community, because these, when somebody's sequencing, it's not a one laboratory, it's a consortium. It's different scientists coming together as a community to solve a bigger problem. So community is a very important aspect of it. I think that's the very reason I have also transitioned into building community, scientific communities and different kind of communities, because a bigger problems can only be solved when like-minded people come together and resolve an issue. So this is a phylogenetic diagram, which is this has been made of all the genome that has been sequenced so far in Microcosm. So let's start with the whole genome sequencing, and the classical genome, first eukaryotic genome, which was sequenced back in 1996. And it was a big, big thing, like sequencing an entire genome, was the yeast genome, Saccharomyces cerevisiae. So it was a haploid set of 16 well-characterized chromosomes ranging from 200 to 2200 kilobases. The sequence chromosomal DNA size, complete chromosomal DNA size was 12.8 MB, and it had 6,183 ORFs and over 100 amino acids long. There was a particular strain which was sequenced, which was S288C. Now there's a dedicated Saccharomyces genome database website as well, where this only and only dedicated to Saccharomyces with different strains, and you can do a lot of study. As I said, plethora of analyses can be done based on whatever question you're asking, or you can download the sequence for learning as well. So the genome was sequenced with a cooperative consortium early 1900s, chromosome by chromosome. The whole genome shotgun approach was not used. It involved 600 researchers, 100 laboratories, and the map was created by all 16 chromosomes with a 10 kb insert in the phage lambda, and the inserts were assembled in the quantic form. So it was a phenomenal discovery, and it made a big news when the first time this kind of sequencing happened. Going back to storytelling, I also want to tell you the time Sanger first sequenced, and to his sheer excitement, the title of his paper was Sequence, Sequence, Sequence. The title of the paper was Sequence, 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 which only shows the level of excitement he had when he first time the sequencing was done, and they had to bring it out to the world and tell that how sequencing is a successful way of doing. Of course, they started with something small. I think 5x174 single-stranded DNA was sequenced through DNTPs and all. But it was a big thing, and that was like the day the thing started, and we are right now talking about genomics. So the comparative genomics is defined as a research strategy to infer the functions and loci of the genome in the target genome from information obtained by analyzing a certain genome, like a reference genome. So what is happening is that you do have a reference genome, and you have multiple genomes which you have sequenced, and you want to do a comparative genomic study. Putting it into simple words, comparative genomics is defined as a comparison of biological information derived from whole genome sequencing. So I would quote a publication which was published recently, I guess in 2020, where 23 Aspergillus species were sequenced. So once you have these sequencing information, what is the next step to do? You align them. The kind of plot that I showed, this can be easily done with the right kind of softwares, where you, of course, you have to have a correct assembly, and all these things, sequences have to be submitted into the repository, and then they are foolproof sequences. And then the next step is that you find out what are the core genomes and what are the specialized genomes which are unique to that species. So that kind of studies were done. So if you see the phylogeny and genome statistics that has been done for this Aspergillus, you will see that all the Aspergillus species are lined down based on the phylogenetic distance. They were, the sizes were put in the form of a bubble. The next is the predicted proteins, how many predicted proteins have been discovered, taking a reference of Interpro, which is one of the repositories of the known proteins. What was the GC content of it? What were the scaffolds, which is information you get once you're sequencing. And then the scaffold has particular cutoffs like scaffold L50, scaffold N50, which gives us that what are the better quality reads that you have used for the assembly. So these kind of preliminary information gives you that why one Aspergillus is different from another's Aspergillus, and what is the uniqueness of that Aspergillus, which makes them for a requirement to be a totally different species. There's some basic monocore genes which make them Aspergillus, but there's certain specific genes which have a specific function, which they've evolved over the time to be a completely different species. This kind of alignment was done through muscle, which is a phylogenetic tree construction tool. In the second graph, this is a dendrogram where we are seeing the phylogenetic relationships between the 29 aspergilli. So here, the green bar shows you basically the white boxes represent the protein families unique in the individual. So there's a bar plot in the green core, which are the number of total proteins which are there. The blue, the turquoise are the core proteins, and the orange are the species specific proteins in each species, in species, which is making it unique for it to be called as Aspergillus particular species, and it has a particular function evolved basically based on the environment or that kind of functionality it had to evolve. So these kind of studies can be done with comparative genomics. So moving to functional genomics. So once you are doing comparative genomics, it's a quintessential question to understand that what protein it's translating into, what is the function, what is the structure. So functional genomics deals with the structure, function, and regulation of all genes rather than a single gene of the genome, and a dynamic aspects of genes like transcription, translation, or protein-protein interactions. Those kind of things can be figured out through functional genomics. So to put it very simply, functional genomics is a field of genomics where systemic approach is applied in elucidating the function of novel genes revealed by whole genome sequencing. So if you see these genome, there are 4 different genes which are expressed, and then the protein sequence have been producing different kinds of metabolomes. And those functionalities can be tested through functional genomics. So a very interesting paper which was there was developing functional genomics platform for fungi. So there are different fungi species, and we cannot undermine the how important fungi are actually. If you think from the point of view of fungi from health perspective, a lot of diseases are caused by fungi which are quite fatal. So study of these fungi can actually improve the health outcome. And if we see from the crops perspective also, there's some big famines have happened because of the fungal infections or infestations. So from food security point of view also, you can say that why these kind of genomic studies have importance. So, but except for few model organisms, you might notice that there is a lot of, when we do a whole genome sequencing, there are a lot of uncharacterized proteins also. Uncharacterized genes are there because there is no reference genome available, something which makes them very unique. So with the help of high throughput sequencing techniques now, more functional characterizations can be we have been able to do. But once we when I say that these are characterized, these are at the computational level. So the validation part, the characterization, gene to gene validation, the characterization has to be done through in the laboratory conditions. So from the genotype to the phenotype, different kinds of things, the study has to be confirmed, the annotation has to be done through laboratory experiments. So taking the example of one of the enzymes in this research paper, they talked about zinc binuclear cluster. It's a nuclear, it has a particular function. But when you see in Saccharomyces cerevisiae, that cluster has 55 genes. But if you see it in Aspergillus nidulans, the same gene the cluster had 300 genes. So what are the ecological adaptations that happens across the species? What is the genomic structure or gene content variation that happens across the species? These information can only come out through functional genomic study. What here they have done is there are different fungi species. Some are plant pathogens, some are animal pathogens, some are human pathogens, some are model fungi. These are characterized into different categories of genes or proteins which are responsible for cellular processes and signaling, some that are in information and storage processing, some which are responsible in metabolism, some which have a general predictive only, like whatever is the regulatory function, and many of them have an unknown function. And this usually is done through a reference database like KEGG, which is known as Kyoto Encyclopedia of Genes and Genomes, where there's an entire repository of all entire different information which is available, and you map them with your reference sequence in question, and then figure out that what are the different cellular genes that are available. And then the uncharacterized proteins have to be further taken forward for study. So there are different ways of doing that. Suppose you are doing a study on particular genes. A simple example for that matter would be like many upregulations and downregulations also happen. So these functional genomics can also be studying for, we can study it for those conditions, like suppose a particular organism performs at different stress conditions with a particular media condition, without a particular media condition. What are the genes which are upregulated? What are the sequence information? How the sequences and genes are turned on and off, and different conditions it is exposed to some increasing dose of antibiotics and things like those, and how the antibiotic resistance is evolved. And those kind of studies can also be done through functional genomics. Moving to structural genomics. Now, you have the genome sequence, the whole genome sequence. It could be from a microorganism, it could be from a metagenome, it could be from a microbiome. Where I say metagenome or microbiome, I will also delve deeper into it in the subsequent slides. But it's not one organism per se. It's a whole consortium of organisms, I would say. Many uncharacterized, uncultured organisms would also be there, because with metagenomics, you are also targeting those organisms who have never been discovered, because they were never a cultured condition defined for them. But that doesn't mean that they don't exist. They definitely exist in that particular condition. So once the genomic sequencing is done, and if you want to go further to structural genomics part of it, so you have to purify the protein and then do some in high quality, the protein purification has to be done. It has to go all the level to the expression part of it, and then the protein in question has to be crystallized, or NMR has to be done. So these initiatives are done so that you delve deeper into what is a important structural function of that protein in question. So we are exploring the unknown biology or chemistry, or we want to find the structure, that what is this structure doing. Once you have available information to it, you do further biochemical validation through functional studies or drug target discovery, or further network analysis or metabolic pathway mapping can be done. So these are usually done, and once you have a sequence, a solid sequence is done, these are deposited into a repository known as the PDB, which is the Protein Data Bank. So protein structure initiative principal goal is making structural information of most proteins readily available for the knowledge of the corresponding genome sequence. So if you want to define it in simple words, structural genomics refers to the exploration of protein structure on a genome scale. So here, as I told in the previous slide also, this is just telling you in words the target selection, cloning and expression into the vector, high throughput protein purification, structure determination either by crystallography at synchrotron source or by an NMR method, and then going further and elucidating the structure behind it. If you have a reference, there's a blast which is known as the PDB blast, which is very, very specific to protein. And then if the protein has a similar kind of fold, it would give you the hit. But, excuse me, many times the sequence information is not known. And there are different ways of determining and predicting the sequences, like the ab initio method, which technically means taking without any reference from the start. Second is homology modeling. If you know that this particular protein has certain similarities, then you can do homology modeling. And third is the threading method, which is also based on the reference sequence. You thread it out and then create a sequence model. And then, of course, this area goes deeper into like alignment of template, and then there's a backbone generation, loop modeling, sidechain modeling. Then the model is optimized with multiple iterations. So there's a lot of MD simulation and MD refinements happening. So this is a very deeper area of structured genomics, which is which can be also explored with the genomics data. So there are different repositories and reference places, like Protein Data Bank is one of them. UniProt is there, SCOP, which is a structural classification of proteins, and then CATH, which is class, architecture, topology, and hereditary. Those kind of classifications of proteins can be done at different levels once the sequence is determined. So moving to epigenomics. So epigenomics is a study of reversible epigenetic modification in a cell, DNA or histone protein that affects the gene expression without altering the DNA sequence. Two of the most characterized epigenetic modification of DNA is methylation and histone protein modification. Now, these two techniques, these two epigenetic modifications are quite common in the eukaryotic system. In fact, these have been seen in the histone modification has been elucidated to play a pivotal role in the regulatory gene expression in filamentous fungi pathogens. Wide range of biological processes of fungal sporulation, phylogenetic morphological differentiation, environmental stress response, biosynthesis, these are taken care by histone modification. Along with that, with the recent methylome studies, it is possible to do which is called as methyl bisulfide based sequencing method, where what happens is that your methyl modified regions are labeled so that you can find out the epigenomics of them. There are established techniques for that, like DNA methylation analysis technique, which I've said, DNA protein interaction analysis, and chromatin accessibility analysis. So epigenetic analysis research can involve studying alteration of DNA methylation, protein DNA interaction, chromatin accessibility, histone modification, and many more techniques, which can help us understand deeper into what are the epigenetic modifications which are happening in our system in question. So this is here an example, not per se of a fungal genome, but I think this is for the human genome, it was, or animal genome, where different kinds of stress is given to a particular cell. Suppose there's inflammation, pollution, drugs, UV radiation, ionizing radiation, electrotransport system, food adulteration, the kind of exposure we humans get it every time. And a lot of epigenetic changes are happening inside at a subtle level in our body. So these kind of changes happen. There are repair mechanisms which are taking place. There are antioxidants which are taking place. But also there is some certain people have also undergo damage in the macromolecules. These epigenetic modifications happen, like DNA methylation, histone protein modification, which leads to impaired physiological functions like cell death, metabolic disorders, age-related diseases, and those kind of information can be studied through epigenomics. There is a dedicated repository for human epigenomics, which is the roadmap to epigenomics, like epigenomic browser. There is other epigenomic information in NCBI. There is the epigenomics per se is studied more in depth in humans, but that doesn't undermine that fungal systems do not have it, and a lot of excuse me. Is somebody unmuted? Can I request the host to mute them, please? Thank you. So going back, that a lot of research is happening in epigenomics in human genome, but we shouldn't undermine that a lot of research is also happening in fungal genomes, where these epigenetic changes happen and shed light on important information. Let's move. Sorry. Let's move ahead. Microbiome. And for this session, microbiome per se. So this is I think my favorite topic of all, because my entire PhD was on genomics and microbiome, where I think it was early 2000s when we got access to this sequencing system 454, and we were still struggling with the reads. The read length were very small, the budgeting had to be done, and a lot of technical know-how was still something which was still establishing. And I got my hands to do research of metagenomics at that point of time. It was frustrating, of course. There were a lot of frustrations at that point of time, but definitely the booming time when this research was just building up and I was also doing it was something very exciting. So as I said, that with metagenomics or with the microbiome or a microbiome research, we are targeting not only single organism per se, we are doing entire population which is there. It could be like a lump of soil, it could be a sweat from a human skin, because there are many uncultured organisms which are present, and we don't even know the culture conditions to grow them. But that doesn't mean that they are not present. They are definitely there. But in fact, 99.9% of them are uncultured bacteria. But once you have that region, you can always isolate DNA from there. There are established protocols for doing that. There are established protocols for isolating DNA from the environmental sample, from sewage water, and whatnot. In fact, there are like dedicated kits now doing that. With that being done, you need to fragment them. Once they are fragmented, you put them into a vector, ligate them, and then there are different ways of doing your research. Either you are doing a culture-based method, because once you put it into a host, which is a culturable host, then you can do some screening kind of experiments. And otherwise, you can also put it into the sequencer following a sequencing protocol. So there we can talk about some universal primers, because you know that we are not talking about one organism per se right now. We are talking about a big mixed population of organisms. So we'll have a dedicated primers, which are 18S primers, which are specific to eukaryotes. For prokaryotes, it's 16S, which are there. So ITS1, ITS2, these kind of primers are there, which amplify your sequences. And then once the sequences are there, you can do further downstream analysis on it. There are dedicated tools, OTUs. Clustering is the first step that you do, which is operational taxonomic units. But before doing any kind of bioinformatic analysis, you have to ensure and QC that your sequences are very, very clean and error-proof. Because with computation, you know that garbage in, garbage out. Because if you start with the wrong sequence in the first place, the sequence quality reads are not good, they are not assembled properly, then obviously the information that you are going to extrapolate out of it will not be a correct information. So that being taken care of, then you cluster them into different operational taxonomic units, OTUs. So these operational taxonomic units are very specific units based on what is very common among those taxonomies. If there is a reference database, it's well and good. But with metagenomics, usually it's very, very difficult. So there are other dedicated databases like Silva, Unite, SSU, RDP, and newer advanced reference databases are also available. Once that is done, you can do further downstream analysis with USearch, Pipecraft, Mother, Chime. We were very lucky that the time I was doing my PhD, the creators of Chime came to our institute and gave us a full tutorial on how to use Chime. So that really speeded up my PhD around that time. And then there are other different specific pipelines like Find Fungi, And of course, the BLAST, GHOST, and that different kinds of algorithms are there. Now, choice of a pipeline is also very important, and it also depends on what kind of questions you are asking and what algorithm in the backend they are using. Sometimes we limit ourselves being a biologist that we don't know the algorithm what is happening behind, but I would always encourage to collaboratively research. Don't be afraid to ask questions, even for that matter, email to the creators of that pipeline, and you can always resolve the problem in that sense. Once the large genomic data is established, you can then further characterize the kind of questions you are asking for. Like, for example, for me, my question in PhD was that, can I find industrially superior cellulase from the soil sample? A material is coming from bacteria or fungi. Can I find that? And with this quest of microbiome research, I was doing it with a plate, which is a culture method where you are putting them into the culture and then putting them into the screening of the cellulose media, and this time seeing that how well or how strong your cellulase is coming from microbiome, and also through the sequencing approach to see that what all diversity and what all wealth is sitting there in that lump of soil. So this kind of research can also be done with the genome sequencing. May I request to mute? Fungal phylogenomics. I think it goes without question that once you have multiple genomes, like the comparative genomics which I was talking about, the next step would be phylogenetically aligning them. So if you see this path of genome sequence here, right here, they have made a genome map after sequencing and then assembling it. There are various dedicated tools to do that. I think Circos is one of the tools to make those circular maps and mapping them. Once that is done, then you're comparing the whole genome with other sequences that are also available in the repository, or you have also sequences multiple genomes to see what is the commonality, what are the core genes, what are the specialized genes which are there, like what are the gene clusters which are present. And based on that, you can also go further into functional assignments. These functional assignments are that across the species, what are the different genes which are doing same kind of functions. They evolve across the species, which are called as the orthologs. And then these are their dedicated, like I said, KEG is one of these. COG IDs are defined, which tells us that these are the functional assignments of these genomes. Along with that information, there's a phylogenetic tree where you can characterize them into different species, what is special about them and what is not. And then metabolic characterizations can also be done. So suppose they were the genes in question were these set of genes, FOLP, FOLK, FOL, and what were upregulated, downregulated, what were the ribose switches happening, and what is unique to different species. That kind of information could be done further in the downstream analysis. So I want to tell again a story about my postdoc when we were doing this patient samples. So this was a disease case of cystic fibrosis. Cystic fibrosis is a disease which is a genetic mutation in CFTR gene. And what it causes is that your lungs are unable to clear the phlegm or the buildup which happens when you're, remember, when you get a very coffee, then there's a lot of phlegm and a lot of mucus which is collected. But you're in cystic fibrosis condition, you are unable to clear that cough, and it just persists in your lungs. And considering that once that mucus is present in your lung, it's a perfect medium for secondary growth of other microbes which might be also there. On top of that, the CFTR people with cystic fibrosis are given a lot of antibiotics to take care of that. And we also know that the more antibiotic we take, the microbial population also evolves further. So more stronger the antibiotic the person is taking, more antibiotic resistant strains are getting developed in your lungs. And more further antibiotics are further given to take care of that, and more antibiotic resistant strains were developing. So our job in my research was to collect the sputum of different patients who are coming to the hospital. And it was a longitudinal study, because once you are sick, you come to the hospital, you get better, you go back, but again you will have a tendency to fall sick again. You'll come back to the hospital again. So every time we are taking the sputum, what was the dose of antibiotic that was taken, and then taking those sputum, collecting the genomic DNA from that sputum, the microbiome of that lung, and then putting it to as well as culture-based plating to find out how antibiotic resistance this particular strain is getting over the longitudinal period of time, to see that this strain is evolving. So it could be a same strain. For that matter, there are fungal populations also which were there, as well as bacterial populations which were predominant. For that matter, Pseudomonas was there, Stenotrophomonas was there, a lot of mucus-liking bacteria, but also a lot of fungal populations were also there. And then we published a very fantastic paper of the how these genetic rearrangements were also happening at the molecular level of these fungi and bacteria. And then they were evolving in antibiotic resistance over the time. And this phylogenomic analysis can be done and dealt upon through phylogenomics. Pharmacogenomics is another area, but this is mainly, I would say, is more with point of view of humans, but I will tell you how it's relevant with fungi as well. So pharmacogenomics is a study of role of genome in drug response. Okay, because pharmaco means drug, and genomics is the genome of the host, which reflects is combining the pharmacology and genomics. So what is happening here is that suppose you are immunocompromised, like for example, in the case of cystic fibrosis, the patients are immunocompromised. Similarly, in the case of cancer, a patient is immunocompromised. When a patient is immunocompromised, then whatever drugs you're giving, and whatever medicines you're giving, so antifungal drugs also you're giving, because your body has fungal infections coming up and things like those. So it is very personalized thing also, because it's based on the genetic makeup of the patient as well, and how they are responding to the drugs. So those kind of studies can be done through pharmacogenomics, where the human genome is taken as a reference, and then you do a lot of drug interactive studies. This could be done through structural genomics aspect as well. So pharmacogenomic deals with the influence of acquired or inherited genetic variation on the drug response by correlating DNA mutations like single nucleotide polymorphism, copy number variations, insertions and deletions, and the pharmacokinetic and pharmacodynamic aspect of it, where how well the drug will be absorbed, what will be the distribution, what is the metabolism, and what is the elimination of that drug once it's given to the patient, or what is the effect which is mediated through the drug's biological targets or the immunologic endpoints. So there's this area of computational, which is known as CADD, computer aided drug design, which is completely dedicated to different drug targets, that how the drug will perform better, is it good enough, which is also an important area in pharmacology, but also with pharmacogenomics it can be applied upon. So this is a very interesting research which I came across. This is from University of Queensland, and it was known as a pragmatic study. That's the name of the study. I'm sure there's a abbreviation to that. And the study was to investigate the benefits of treating cancer patients with tailored doses of antifungal medicines. So the pharmacogenomics aspect was used for better treatment of fungal infections in cancer. This study has just recently been started. I think it's funded for like 3 years by the Medical Research Future Fund, which is in Australia. But I just wanted to bring this up to tell you the different avenues of genomics that are there in like what are the different avenues one can take with the study of genomics. And right now we are talking about fungal genomics per se, but it can be extrapolated into different organism system of your choice as well. pathogenomics. We know that fungi can be a big cause of pathogenesis. A lot of crop damages happen. I said that a lot of diseases happen, which are sometimes fatal. And especially in the case of immunocompromised patients, it just gets way too fatal. So pathogenomics is a field in which you use high throughput screening technology and bioinformatics to study encoded microbe resistance. Right now, I'm talking about microbe, but this can be extrapolated to fungi as well. In virulence factors, different, what are the virulence factors which are present, which are very specific in causing the pathogenicity? How can we turn off those pathogenicity areas? Something which is known as pathogenicity islands in the genome sequencing, which can be determined, and which enables organisms to infect the host and possibly cause the disease. So these genomes of pathogens can be also, it includes studying genomes of pathogens which cannot be cultured outside the host. So we are, with the advent of genome technology, we know that we are not dependent on the cultured microorganisms, as well as the uncultured can be deciphered, information can be taken out from there. So pathogenomics has also allowed researchers to better understand the genome evolution per se, the different why a particular species, a particular strain became pathogenic. What was the gene loss? What was the gain? What were the duplication? What were the gene rearrangements which were happening? So these kind of studies can be done at the level of comparative genomics, where the same species with different strains are lying down, and you see that what is making them more pathogenic or pathogen and ability to cause diseases. Similar to that, how can we upregulate the immunity of the host as well? So in one of my projects, when I was doing my postdoc, another story time, of course, is we were studying the host pathogen interaction in plants, plant pathogen interaction, where the fungi was infecting the plant, which one wouldn't desire, because it would create a lot of crop damage and things like those. But they were also studying the pathways of immunity of the plant, how to make the plant more immune. So when we study immunology, we always study from the perspective of animal immunological systems, the whatever is the T cells, B cells, and things like those. But from the plant immunology point of view, the studies were going on that how to make those plants pathogen resistant, and how can we, and for that it's a both way study. One group will be studying the host, another group will be also studying the pathogen aspect of it, and what are the targets which can be done to obviate those pathogenesis. So those very interesting kind of studies were happening in Goodman Lab when I was studying there, and you learn different aspects of it. So Patho Systems Resource Integration Center is one of the resources where one can have a repository of different pathogenic related genes and information related to that. I'm not sure it's very specific to fungi per se, but it has every organism information over there. We also have a pathogen watch. There's a virulence factor database, VFDB, for pathogenic bacteria. And there is other databases for human and animal pathogens. I'm sure there are more dedicated resource databases for fungal pathogens. If you are aware, you can definitely tell me. Probably somebody from Mycoasia can tell me if there is. If not, then definitely there's a scope of creating one, which also is a very positive thing in research. The next area which I'm going to touch is metabolomics. So metabolomics is a scientific study of the chemical processes involving metabolites, small molecule substrates, intermediates, and products of the cell metabolism. It trickles down from the whole genome sequencing only. Bear with me to see this. So obviously, whenever you have a particular sample, right now this is a fungal sample, you will be preparing the DNA. This will be translated into RNA and then the proteins. Those proteins can be then further identified into what are the metabolites which are produced. by those proteins. So once those metabolites in question are there, giving the right conditions, of course, that these kind of metabolites will be produced, giving whatever is the study question in mind, like whatever stress condition you're giving or different environmental conditions you're giving, to see that what are the metabolites or the metabolomics of per se of that system. This can be done through techniques like NMR, mass spectroscopies, where the data is acquired, noise filter is done, data processing is done, and then downstream normalization, peak detection, principal component analysis, then volcanoplots, and then heat maps are done of what are the different metabolomes which are produced. And then that information can also be taken forward and shedding light on the what kind of metabolomes are contributing in that. So I think that I finished way before time. I underestimated, and probably this gives us more opportunity to discuss as well. I will say that fungal genomics has a lot of implications, a very, very powerful system. And back to the one more very interesting story about fungal system would be that how old is this fungal system? It's as old as 1940s, when Beadle and Tatum did this experiment with Neurospora crassa, because the bread molds, the Neurospora crassa, is such a easy system to work upon. They did mutation by exposing with chemical radiation, and the effects of mutations were then studied, and then the classic hypothesis, one gene, one enzyme, was given. The reason being that fungal systems have a short life cycle. You can study mutations and those systems. It's relatively easy to manipulate, grow, and the laboratory. The genetics is pretty well established. The fungal system genetic information is very, very deep, I would say. To do more understanding, is quite versatile. So based on that, a lot of studies, and I think the fungal system is very conducive to deeper research. In fact, there are a lot of genomic similarities between fungal genome, and it can be extrapolated to humans as well, with the drug-related studies, with identification of new drug targets, to understand the fungal biology per se, for new diagnostic tests, even the fungal ecology, because soil health is another area of research which is happening nowadays, because our soil is also depleting its nutrition. So the fungal ecology aspect, there are dedicated groups working on that. And there are new bio-based products solving bigger problems like plastic pollution and things like those, or different problems can also be like extrapolated through the fungal genomics. And that's how I, with a heart full of gratitude, I thank you all for paying attention to this seminar. I ran a bit very quick, so there might be many questions. We can definitely take few questions. I'm just giving a heads up, I'm not a fungal expert, and if there's somebody from my ecology team is also there, they could also respond to the questions. I'm more into the genomic side of it, and I've worked with those kind of systems. But I apologize if I finish it a bit sooner, but it gives you more opportunity to pump up with the seminar and probably dig deeper and pick up a research paper. There's a dedicated research in fungal genomics per se, the technicalities, the techniques, any area you choose. You can just go deeper into it, and the sky is the limit. Your favorite area could be the comparative genomics, it could be functional genomics, it could be structural genomics, where the MD simulation is a deep topic on its own. And there are other areas, like I've talked about pharmacogenomics, phylogenomics. There's a dedicated lab working on only phylogenetics, and like that. So thank you very much to the bioinformatics team for giving me this opportunity to just give you a glimpse of the tip of the iceberg. And we can always have a dedicated session on each topic and study more about it and learn more about it. So thank you, ma'am.
Thank you so much, ma'am. I would like to request the participants to post the questions in the chat box. or they can turn on their mic and ask directly, whichever is comfortable. Let's open the session for question and answers.
So if they are asking question, I would request the host to take a name so that that person only ask the question. So there's no so there is no chaos in like two people asking the same question or the cross communication happening.
Like we have a very first question from Sivaraj Irusapam. What is the best and cost-effective kit for the fungal library preparation?
I think that there are many because I'm off market right now, so I wouldn't be able to say top of my head. But there are a lot of library kits. In my time, it used to be Kaizen had a very nice kit, and there were newer brands coming up as well. So I wouldn't be able to say the brand name per se. I wouldn't be promoting a commercial brand right now. But since I have my email address right here, please note it down, and then please have a follow up with me, and I'll definitely guide you with the right kit, which will be in budget for doing research in a small scale in India for fungal isolations. When you are saying fungal isolation, are you saying the single genomic isolation, or are you saying with microbiome kind of study?
And we have another question from Som Subrata Datta. What is the scope in mycorrhizal genomics?
Oh, that's pretty promising because mycorrhizal genomics has a lot of application. A lot of startups are coming up with these kind of areas. And if you look into research papers, there's a lot of work happening in that also. I'm not sure how easy it is to isolate the genome from there. One has to delve deeper into it and see that what all research papers are there, what are the established labs, what are the troubleshooting caveats they are facing, and how established it is. As I said, that I'm not a fungal expert to comment much on that, but do email me and I'll definitely follow up and give you right papers if you need, and then we can take the conversation forward from there.
One more question. How does fungal genomics contribute to the discovery of new drugs or bioactive compounds produced by fungi?
See, so of course, these fungus is a very important system. There are a lot of important metabolites this are discovered, but downscale you have to also I think that scale it up. So I know that in my laboratory itself, we had this uh, in my PhD, we had this work on streptokinase. Streptokinase is a drug which is here, please. Streptokinase is a drug which is used for our atherosclerosis, which is your blood clotting in the bloodstreams. Now what is happening is that you are going to create a drug for that, but if you produce it in mass scale, like you have to do a fermentation and scale it up to the industrial level, then the system that you're using would don't do justice, because a lot of things would work better on a fungal system. So the vehicle which was there was changed to Pichia system. Pichia is a fungi, and there the drug was again synthesized, and then seen that what all modifications uh, streptokinase has to go through, and then they were better for the mass scale production and scaling it up into fermentation department. So uh, many drugs which are good for human body would work better on a fungal system. Then you have to scale it up in fermentations level.
Okay, one more question now. Metagenomics is the study of the genomes of many organisms at once, correct?
Yes.
Then wouldn't it be confusing as we wouldn't know which sequence comes from which organism?
So that's why I said that when you have many multiple sequences, you have to then do the downstream processing of it. So that can be only done through dedicated tools like Chime. where what happens is that once you have entire DNA sequences and then the assembly is done, there are certain core sequences which gives them the particular identity that this organism belongs to this particular species. There are core genomes which give them the identity. Like I said that, how do you do taxonomy? You do taxonomy because there are certain sequences which give them the uniqueness in identifying that these particular sequences belong to that particular species. So once you have this mix of sequences, even those sequences can be separated out based on the uniqueness in it. And then phylogenetic analysis can be done to determine that how they are diverging in that particular system, or what other functionalities they are holding in that particular area, or what is the role of those microorganisms in total, what they are doing. So those kind of things can be done through metagenomic tools which are available as well. It is, as I said, that every topic is a topic on its own, and when I'm speaking about it right now, I'm speaking at a very preliminary tip of the iceberg level, because there's a lot of nitty-gritties and what of technicalities that needs to be discussed before one gets into it. So that's why I said that if you want some papers, feel free to search or email me at what kind of problem question you want to address, and then there'll be dedicated papers I'll be happy to give you, and we can take the conversation forward from there. So primer designing is something which is also something very specialized that people who are regularly doing wet lab would be able to guide you, because I have not much into that area right now, so I wouldn't be the right person to help you with designing. It's also a session which can be taken definitely in future.
Okay. There's one more question. Are there any ethical or environmental considerations related to fungal genomics in terms of genetic modifications?
Actually, there is, because there is a lot of ethics has to be taken with the advent of next generation sequencing. Sky is a limit. You are doing a lot of sequencing, a lot of these CRISPR-Cas system has a lot of modifications happening. What is the downstream impact of these things, and what are you giving out to the environment? Would be something which would be quite concerning, because you wouldn't want to have a new super bug created out of it, with some modifications, some mutations, or those kind of things, and then giving out and throwing it out into the environment. So those kind of ethical issues are there. And then with the human genomics, the data, the kind of data which is there, you cannot just give it out for commercialization and those kind of things, because one could make money out of any information as well. So for science purpose, yes, everything should be given out for research, for the growth. But when you're thinking about commercializing particular things, in earlier form when Human Genome Project was there, a lot of scope of commercialization, personalized medicine, precision medicine, these kind of terms were coming up. But for ethical standpoint of view, everything was kept open source, so that nobody could make a money out of somebody's weaknesses, or make something out of it. So one has to be very, very ethically compliant. Right now, India is still at infancy when we say about these kind of genomic researches, what ethical compliances have to be taken care of. But I think that those policies also have to come out over the time. And probably there are some policies with the genomic research which should be out when somebody is doing genomic research. So declarations like those are also equally important.
Ma'am, there is one more question. How is myco-endophytic genomics contribute in its prospecting studies?
This would be very, very specific area, I think, like myco-endophytic genomics, mycorrhizal genomics, phytogenomics. It's that the system is changed, but of course, it depends on why. The why has to be clear. The person who is picking up a particular system, why did you choose that system? The question answer lies with the person who started the research on that. So any system, whether you take a bacterial system, whether you take a fungal system, establishing the right question at the first step of it, to extrapolate into what all scope it has and what all areas it can go deeper into, and what are the possibilities with it. These things are also based on the innovative ideas what a scientist brings forth based on the already established systems which are doing there. So when you ask a question that you pick a very particular system, that mycoendophytic system or mycorrhizal system for genomics, is it good, is it bad? The answer lies exactly on why did you choose that system. So the question goes back to the person who's asking, because why was that system used for genomics? And that would shed light already on the kind of scope it has. Of course, you can do some dedicated bibliography research on the publications which are happening in this area, and then it will give you more light on what all scope it has and what all possibilities it has in doing something. Possibly there's a biofuel possibility, there's soil replenishment possibility, crop improvement possibilities, and those applications will only come because that's your particular system. So when you pose a question to somebody, that person might not know all the systems of the world, right? There are different systems, there's so many systems, bacterial system, fungal system, algae system, and every organism can be sequenced. Even a speck of dust can be sequenced, right? And then people have published papers on a speck of dust, that wow, what all diversity is there in a speck of dust. So the person who is initiating the research would have the right answer into why there is a need of genomics also for that system. Every system need not be sequenced just for the sake it has to be sequenced. The question has to be very, very strong and valid. And based on that, when in the future, when people write project grants on why research, I want to do a work on this system and I want to sequence them. Based on that sequence, I want to do these kind of questions that I'm asking. Then the government grants a particular amount, a lump sum amount on this is the research grant dedicated for this. There also, there has to be a proper bifurcation of how much money is going to go on the sequencing, how much is going to be on bioinformatic analysis, how much is going to be in a cross-validation in the wet lab. So I think that when somebody is asking us a question specific to a particular system, the answer lies by the person himself, that why the person chose that system. Thank you.
And just one more last question. Can we use the same fungal sequencing technique for lichen also, since it contains algae and fungal partner?
Sure. The protocol might be different. There might be established protocols because every system has a different way of extracting the DNA. But it can be always extrapolated into different areas. Of course, it can be done. So lichen is a different system. In fact, I was showing about the database of mycocosm. So there's also a special database called phycocosm, which is only dedicated to the algae system, where there's a whole repository of algae which are sequenced so far and what all bioinformatic analysis you can do. I would definitely urge you guys, maybe I should move to that slide and show you that slide. Yes. So the JGI is a mycocosm. They also have a phycocosm, which is very dedicated to algae system. So they're dedicated pre-sequences also, which one can download and then study, ask the question, take it as a reference, do comparative genomics, and then probably sequence their genome from their source and then put it out there and then say, compare it, that what are the subtle differences which are there in the system, which is already present in the data repository versus what is novel and unique to your organism of choice. So those kind of studies can always be done.
One more question. Which sequencing method has been used for fungal sequencing?
Sequencing, see, it depends on if you're doing genomic research, then a lot of Illumina-based sequencing is done. This is basically you extract the entire genomic DNA, then you fragment it, put it into the library. But newer methods have also been available, like the nanopore-based method or iron torrent methods. And the sequencing has also, the sequencer per se, the next generation sequencers have also established a lot, evolved over a time. to give a longer reads, better reads, because you have to fragment them to put it into a vector. You have to amplify them, and once you have amplified them, what you come out, you have to QC them. Once the QC is done, those contiguous repeats have to be assembled together in the right form to form the scaffolds. Those scaffolds have to be then further QC'd to see that how much is their N50, or what is the quality of the reads to take it forward for assembly. So over the years, a lot of sequencing has also evolved with the sequencing technology, and then based on that, more established methods one can choose. But I think that Illumina has been established for pretty many years now, and Iron Torrent has taken over, the nanopore method has taken over, and newer technologies have also come forward. I think I noted it down. It was third generation. You have those single molecule sequencing. This is a newer form of sequencing, which is more robust, it is said. Then there's packed biosequencing, which is there, which was from Helicos Biosciences. So newer technologies are also coming, which will give you a longer reads, more error-proof reads. If you see one of those thing which I was showing about the history, you will see the simplicity to complexity. Early 1965, very simple organisms were sequenced: E. coli, phage, viruses, and all. And as we move further and further, 2000 onwards, more complex systems, more like they have more repeat sequences. Those kind of sequences have also been able to be sequenced, like Arabidopsis, human, mouse, dog, rat, chimpanzees, genomes of zebrafish. And then there are like plants, and a lot of a lot of anthropology related researches have also taken place, where like whatever were the bone reserves from the caves of ancient Neanderthal or Denisovan, they were also sequenced, and then information could be deciphered how they were genetically different from the modern man. So this also depends on the newer sequencing technologies that are present, which will give you more robust, longer reads. Like I said, that the third generation reads, which are available now, are the best ones.
Um, I think there are no questions, so we can proceed towards the valedictory part.
So thank you very much. And if you have any further questions, feel free to email me. I have my email ID here, and we can always solve a problem together. Thank you so much.
Thank you so much, ma'am. I'm Mr. Abhishek Das on behalf of Patkar-Varde College and Department of Bioinformatics. Feels heartful of gratitude and admiration for the remarkable journey we have undertaken together in this enlightening webinar on fungal genomics. It is truly an honor to address such an esteemed gathering of individuals who are deeply committed to advancing our understanding on fungal organism and finding biologically meaningful insights. I would also like to express my profound appreciation to Dr. Bele Damodara Shenoy, co-founder MycoAsia, and our speaker, Dr. Vijaya Brahma, who generously shared her expertise, insights, and breakthrough findings. I begin my vote of thanks by addressing our appreciation to the management of Chikitsak Samuha, Chief Education Officer Dr. Mala Kharkar ma'am, our principal Dr. Pratibha Gaikwad ma'am, Chief Coordinator of Science and Coordinator of Bioinformatics, Mrs. Ruchita Dalvi ma'am, and to our sponsoring organization, RUSA. I extend my heartfelt thanks to each and every participant who joined us from around the world, bringing with them a wealth of knowledge, diverse perspectives, and unwavering dedication. Your presence here today underscores the collective determinations to decode one of the most important aspects of fungal genomics. Behind every successful webinar is a team of dedicated individuals who work diligently behind the scenes. My gratitude goes out to the teachers who ensures the seamless execution of this event, allowing us to come together in a virtual space where ideas and knowledge flow freely. As we depart from this webinar, let us carry forth the torch of collaboration, innovation, and empathy has illuminated our discussions. Uh, Once again, thank you, everyone. The feedback will receive, the participants will receive their feedback participation certificates through mail once they fill the feedback form. The link for which has been posted in the chat box. Please note the link will only be active for 30 minutes. So I declare the session as closed, and I want to thank each one of you once again for your unwavering dedications. Thank you.